Hello, today we're going to be looking at mitosis. Mitosis is the process of cell division in somatic or body cells, and its three main uses are growth, repair of damaged cells, and asexual reproduction. So, cell division is put into two different categories. So this is interphase, and this is mitosis. Mitosis, where the cell actually divides, is just a tiny sliver. Interphase, where the cell prepares for division, is where the cell will live for most of its lifetime. So this is the G1 phase, where the cell duplicates its organelles. This is the S phase, where the DNA synthesizes, and this is the G2 phase, where the cell checks to make sure that these two phases were done correctly, and if everything is good, then the cell will go into mitosis. So this is what a typical cell looks like in interphase. We see the DNA is uncondensed here, the nuclear envelope is still intact, and we have our organelles. So this is prophase. We see here the DNA is condensed into sister chromatids, and our nuclear envelope is starting to disappear. That's the first phase of mitosis. This is metaphase, the second phase. We have the spindle fibers that originate from the centrosomes. They are attaching to the kinetochores of these. They will attach to the kinetochores of the sister chromatids, but for right now, the sister chromatids are just lined up in the middle of the cell. This is anaphase, where the spindle fibers attach to these kinetochores and start to pull them apart. This is telophase. We see the DNA is already starting to decondense. The nuclear envelope is starting to reform, and we have these two cells that are starting to pull apart from each other. Lastly, we have cytokinesis. The two daughter cells are formed with identical DNA, and mitosis is complete. Thank you.